In this video, I will be discussing some post-translational processes that are happening to the polypeptides that enter the endoplasmic reticulum. Okay, so just a review, the endoplasmic reticulum is the factory wherein the proteins and lipids of cells are made, at least a majority of them. So the proteins are made by the ribosomes, which dock onto the rough ER, which then deposit the nascent polypeptide through the translocon so that it can enter the lumen of the rough ER. Okay, the lumen or the cistern of the rough ER. Okay, so now let's talk about that processing. That processing is generally the addition of carbohydrates to one end of our nascent polypeptide. Okay, so what are we looking at here? Here we can see that ribosome depositing the nascent polypeptide. And look at the final product. Okay, in the final product, there is an attached Christmas tree-like structure that we see here. And if you look at the legend, these triangles, diamonds, and circles are actually sugars. Okay, so that's what we're gonna go through in this illustration. Okay, so first, we have a molecule called a dolichol phosphate. Okay, the dolichol phosphate is a resident molecule in the membrane of the rough endoplasmic reticulum, meaning to say it's always there. Okay, and take note what's happening here. Through a reaction from uracil diphosphate. Okay, so this is uracil diphosphate. So very similar to your ADP in structure. So uracil diphosphate that's carrying N-acetyl glucosamine. Two of those are gonna be reacting with the dolichol phosphate, and it's gonna cause the reaction. The reaction is gonna cause the addition of those two N-acetyl glucosamine or NAGs onto your dolichol phosphate, and at the same time making it a diphosphate. Okay, and then next, in step number 2, we have another molecule that will react, this time GDP, to add mannose sugars, and that's 5 of them. Okay, on top of the NAGs that was added here. Okay, so GDP is gonna be adding that to our ever-growing Christmas tree. So here we can see the 5 mannoses. Okay, and then somewhere here in step number 3, our dolichol phosphate with the NAGs and mannoses are gonna be flipped. Okay, it's gonna be flipped so that the sugars, instead of facing outside into the cytosol, it's now facing into the rough endoplasmic reticulum lumen, as we see here. Okay, so that's step number 3. And in step number 4, there's a series of dolichol phosphates also that is carrying with them more mannoses. Okay, and then they will continue on flip-flopping, facing the outside and then facing the inside, and that will take 4 of them. Okay, because it's gonna be carrying one mannose one at a time. So we can see what's being accomplished here. It flips back down when it gets the mannose from the GDP. Okay, and as it flips down, it actually attaches the mannose to this ever-growing tree of oligosaccharides. So you can see the final product here. So you can now see there are now more mannose molecules. Okay, and then here in step number 7, there are also more dolichol phosphates. So you can see the importance of dolichol phosphates here that are getting glucose molecules from UDP, so 3 UDPs. And these dolichol phosphates with the glucose are also going to flip down into the lumen, and as this ever-growing Christmas tree of oligosaccharides passes through this phase, those glucose, 3 of them, are gonna be added to form now these 3 glucoses that are added here. Now, very important, don't forget about these 3 glucose molecules because they are going to be used later on as a checkpoint as whether your nascent polypeptide is correctly folded or not. And then finally, a reaction is going to transfer this, it's going to cut this portion just under the first nag and transfer this Christmas tree of oligosaccharides onto your nascent polypeptide. And voila, you now have a nascent polypeptide with one end full of oligosaccharides. Okay, so that's the glycosylation process that happens in the RER. And then your dolycol phosphate becomes a dolycol diphosphate, and one phosphate is going to be removed and the cycle repeats itself. Okay, so what is the quality control for misfolded proteins? So I told you to remember the 3 glucoses that was attached to your polypeptide in the previous process, in the glycosylation. So here, we can see an enzyme, glucosidase 1 and 2, which are going to nip off 2 of these glucose. These glucose molecules, so that you will just result in this polypeptide here, okay, with the glucose. And then this polypeptide with the glucose is going to be checked by calnexin. So calnexin will only recognize one glucose molecule. That's why this step, in step number 1, the cleaving of the last 2 glucose molecules is very important. Because calnexin here can only detect or bind to one glucose molecule. So we can see calnexin is bound to the glucose, which is bound to the other oligosaccharide, and to the protein itself. And calnexin checks whether the protein is folded properly or not. Okay? So, if not, let's move on. If not, or if it has folded properly first, then it's gonna be sent. This is when it's not folded properly, it's gonna be sent back into the cycle so that another enzyme, UGGT, can add 3 more, or can add this glucose back into that polypeptide. Okay, because calnexin is gonna remove the glucose. The glucose is like a ticket, you know. When you enter the cinema, the ticket will be torn. So if you will go out and then you want to re-enter again, you need another ticket, and that's being provided by UGGT. Okay, that glucose ticket. So it's given another chance to fold properly. UGGT will give it a second ticket. It's gonna go back to the calnexin to test for proper folding. If it still fails, calnexin will let it go back to UGGT to get a ticket and to fold properly. There's a limit though. Okay? There's a limit because what calnexin actually gets is not just the glucose, but also a portion of the other oligosaccharides here. So calnexin will recognize an ever-returning misfolded polypeptide and will say, you've returned 10 times already and you're still unable to fold. So I'm going to send you now somewhere. And that somewhere is to number 7 and number 8. So the polypeptide will be sent to a proteasome. And this is an enzyme that breaks the protein apart, that destroys it. So misfolded proteins are destroyed. So only then when the protein is folded properly will it be sent into the exit, where the exit is in a form of a vesicle so that it can be carried now to the next step, to the Golgi complex. So I hope you understand this process of quality control for misfolded proteins. Okay, so there is this mammalian unfolded protein response. Okay, so we have here our sensors for these misfolded proteins. Okay, so So in the first sensor, we have the PERK proteins, which actually later on become a dimer. Okay, so what happens if there are misfolded proteins? These BIPs are actually, they actually help the protein fold. Okay, they are chaperone proteins. And when there are misfolded proteins, they leave the PERKs behind. And the thing is, these PERKs become active when the BIPs leave them. Okay, so I hope that's understandable. So when the BIPs detect misfolded proteins, they leave the PERK. The PERK becomes activated. What happens when it becomes activated? It forms into a PERK dimer. Okay, which will then enable the activation of a translation factor called ELF2 alpha. Okay, and once ELF2 alpha is activated, it goes to the ribosome and it stops the ribosome from doing additional translation of proteins. So meaning to say, stop muna ang production ng proteins because we have proteins here to fold properly. So stop first. So you see the reaction there for the PERK2. Okay, that's the PERK pathway. Okay, next, we have this ATF6 pathway. Okay, so ATF6 is also attached to these chaperone proteins, BIP. And the same mechanism, the BIP prevents this from becoming active. When the BIP detects misfolded proteins here, it leaves the ATF6. The ATF6 becomes active. And when it's active, this cytoplasmic portion is gonna detach from the membrane-attached portion. Okay, it's gonna go up as we see here. And where will it go? It will go inside the nucleus, and it will promote the transcription of mRNA that will be translated to produce proteins that alleviate the stress of the unfolded proteins that are accumulating in the ER. Okay, so that's also the ATF6 pathway. So I hope you understand these two UPR pathways in the mammals. So right now, we have finished discussing at least a general discussion on what's happening in the ER. Okay, we are now going to move on to the next video, the Golgi complex. Okay, but before that, there is a region between the ER and the Golgi complex called the endoplasmic reticulum Golgi intermediate compartment. Okay, and this is where the traveling products from the ER are before they reach the Golgi apparatus, and that will be in the next video.